All right, so we are going to walk through how to do this Western Scientific Method lab. I'm here at home, so if there's dogs or kid noises, please ignore them. But what we are going to try to do today is to be able to make clear and testable observations. We're going to learn how to formulate hypotheses, identify independent and dependent variables. We're going to design and conduct a controlled experiment. And then we're going to figure out how to take that data that we collect and make it into a line graph so we can interpret our results, and then base our conclusions on that evidence that we collect from our data. As you guys have watched in those videos by now, we're familiar with the Western scientific process of make an observation, develop a hypothesis. But one thing that we didn't really discuss very well is the independent and the dependent variables. An independent variable are the factors that are being tested. It's the thing that we're manipulating. In my setup today, my independent variable is going to be different temperatures of water. Here I got some hot water. This is just lukewarm water. And then I've got some water in the freezer. So these different temperatures, treatments are going to be my independent variable. I'm testing the effect of temperature on the rate of diffusion. So then our dependent variable is the thing that we're measuring. It's the response variable. So in this case, we're going to measure how quickly our food coloring is going to diffuse in these different temperatures, right? So our dependent variable, how fast that food coloring is going to diffuse, depends on our independent variable. There's other factors that we need to control for, so other things that might affect how fast this food coloring might diffuse. So what we're going to do is I have the same size of mason jars. You might not have mason jars. You might have some other glasses that look the same. You can use clear plastic. The only thing is that we need to be able to see through these. So as you're going around your house trying to figure out what you have, just make sure that you can see through. Okay. So our shape and size of container is going to be something that we're controlling for, a controlled variable. We're going to make sure that that is consistent across those three different treatments. The amount of water that I have in here, I have 1 cup of water. That's going to be the same across all my treatments. So that's another thing that I'm controlling for. Our quantitative data that we are going to measure today is going to be the amount of time. It's our dependent variable. For how long it takes our food coloring to diffuse completely in our different temperature treatments. Okay. So if we don't have a thermometer, then what we're going to do is have hot, room temperature, and cold water, and those are going to be qualitative data, right? We're not measuring the temperature, but we can say, oh, this temperature is definitely greater than that one, and that one's definitely greater than that one. We can rank them, right? We can have categories, and that's an example of qualitative data. So what I'm going to do now is start to set this stuff up. So I need to have food coloring. I need to have my jars. I've got one already in the freezer. So I've got a room temperature, and then my one in the freezer is going to be for my cold. I have on my jars, actually have these measuring lines. So I just fill this up into the 8 fluid ounces, which is 1 cup. I'm going to have my phone to act as my stopwatch. And then I need to get my labels, because the most important part of science is labeling. All right, so I'm going to use these sticky notes to label my different treatments. So I have hot. Okay, it's my hot treatment. So my room temperature treatment is my control treatment. This is the baseline, but which I'm going to compare my other 2 treatments. My one experimental is my hot, and my other experimental is my cold. I'm going to keep leaving it in the freezer until I'm ready to go so that it will get as cold as possible. Okay, but there's my label. We're going to need to make sure that we have access to that spreadsheet. If we're looking at the digital copy of the lab, then we can access that class data spreadsheet straight through this link. It's also linked in Canvas. Okay, so when you access this, you're gonna come right here to File. You're either gonna download this as a Microsoft Excel, or if you want to keep working in your Google documents, you're just gonna select Make a Copy. Please do not request access. I get a million emails when that happens, and then I get grumpy, and you don't want me to be grumpy. Okay, what you're gonna do is you're gonna change these to match your experiment. Okay, we're going to do this experiment 3 different times. All right, I'm going to set up my treatments. Got my hot, coming in with my electric kettle, filling up to my 8-ounce mark. It's about 200 milliliters on your side. Nope, got to get my room temperature water. Just getting this from my sink over here. Cold. Got my nice ice water. Gonna strain it. Keep the ice cubes out, but let the cold water in. Let's see. Start cold, room temperature hot. Once I put that, and I'm going to give each of these a stir using my super cool stirring device, which happens to also be my thermometer. I'm going to go do this quickly. 1, 2, 3. 1, 2, 3. 1, 2, 3. Stir, 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 and stir. Now it looks like my hot's already done. So that was like 7 seconds. The cold looks like it's getting there, but I can still see. Yeah, I'm going to call it for both of those. Now I'll do this 2 more times to get my 3 replicates.